All right, so all we defined so far with proteins was amino acids, but there's something called levels of protein structure. Amino acids can link together. What's important is that they fold correctly and form the correct protein structure. Literally just link a chain together, and the chain folds based on how all the functional groups fit together. How do they fold? Well, we first have to know the levels. There's 4 levels. The first level of protein structure, what's the amino acid sequence? There's 20 of them. Which one and in what order? Okay, because you're forming a singular polypeptide chain. Amino acid sequence of polypeptide chain, a chain of amino acids. Peptide because peptide bond links them together. So just imagine a string of pearls, just kind of, each pearl is an individual amino acid, and what is the amino acid sequence from beginning to end? Okay, that's it, just the sequence. That's the first level, the correct sequence. If it's not the correct sequence, it's not going to fold correctly, and usually you'll get a non-functional protein. So the second level is the simplest structures of the folding. Okay. So the second level, the amino acids will start to fold in a way in either 2 basic shapes, alpha helices or beta pleated sheets. That's kind of the name of it. Alpha helices or beta pleated sheets. It's like the helices, they kind of fold in a way in a helix. And there's kind of weak bonding between the amino acids in this structure here to kind of maintain this structure. Whenever they show a dotted line, that usually symbolizes some sort of weak bonding, like hydrogen bonding, and that's what it says here. Spiral structure stabilized by hydrogen bonds. And here you can see the hydrogen bonding forming this pleated sheet. So I'll just kind of note stabilized by hydrogen bonds. Stabilized by H bonds. So a pleated sheet is just kind of a rigid sheet structure. And so the sequence can fold one way or the other into either a beta pleated sheet or alpha helix. And this polypeptide chain, it's really long. And it could be a series of helices and beta pleated sheets in a row. And so that's kind of like the third level of organization, the tertiary structure. So tertiary, I'll put third instead of tertiary. It's primary, secondary, tertiary, quaternary. So third is tertiary, and you have many alpha helices and beta pleated sheets in a row in a single polypeptide chain. Many alpha helices, beta pleated sheets in a row, in series, one after the other. They're continuous. So maybe you have alpha helix, continuous with the beta pleated sheet. Maybe you have another alpha helix, maybe another beta pleated sheet. But there's a beginning and there's an end. There's a start and there's a finish. The point is, it's one polypeptide chain, this tertiary structure. And maybe there's weak bonding and that kind of It kind of maintains that shape, right? That's kind of the definition of it. So the quaternary structure, I'll put fourth level of organization, also known as quaternary. This level of structure is more than one polypeptide chain linked together. Let me read from the slide. And two or more polypeptide chains, each with its own tertiary structure, combine to form a functional protein. Two or more polypeptide chains linked together. By stronger bonds, like for example, disulfide bonds. Two or more polypeptide chains linked together by disulfide bonds. So let's get another one of these polypeptide chains. And then there's another one, and they're linked together by a disulfide bond. Very strong bonds. So that's the disulfide bond there in red. They're strong bonds. When you work with proteins in the lab, you have to break those bonds if you want to run them on a gel, let's say. And you need really strong chemicals. I remember how bad these chemicals smelled. It was beta-mercaptoethanol. It was super toxic. It smells super bad, but that's what it takes. Super strong chemicals to break those bonds. They're very strong. Okay, primary, secondary, tertiary, quaternary. Know that. On your little half sheet, you've got some space. Squeeze in. We did two homeostatic ones, right? And that was like 1 to 6, 1 to 6. Why do you call this one number 12? Okay, that's what I'll look for. And just put 12A, 12B, 12C, and then identify based on the picture. What do you think the level of organization is in the protein structure for 12A, 12B, 12C? Do it on your half sheet now. Would you want another one? All right, that was enough time for you to guess on some of them. What do you think A was? A is not primary. A is secondary because they're illustrating an alpha helix, which is the second level. Okay. What do you think B is? Knowing what A is. B, shown here, is a bunch of alpha helices in a row. That qualifies as tertiary. Okay. Because it's still one polypeptide chain, but it's many of these secondary structures in a row. Therefore, what do you think C is? Quaternary, because it's many of these linked together. That's why they use the color code. So this would be a tetramer. There's 4 polypeptide chains linked together. Okay? All right, so that's level of structure. Yeah?
One more time with A being secondary. Can you explain that one?
Yeah. The definition of secondary structure is it forms an alpha helix or beta pleated sheet. So that's an alpha helix, so it qualifies as secondary. Okay?
I see.
Moving on. Bonding. More chemistry. So I'm done talking about carbs, lipids, proteins. This is another chemistry topic, bonding. Be kind of quick about it because it should be a review as you've taken chemistry. All right, an ionic bond as shown here involves the transfer of an electron. Electron negatively charged molecule. So if chlorine is kind of given up that, from sodium to chloride is the transfer from there to there. What results is a fully charged sodium and chloride ion. So that fully positive sodium and fully negative chloride ions, there's an electrostatic interaction between the ions that form. Okay. Sodium is a monovalent cation, and the chloride ion is an anion, fully positive, fully negative charge as a result of the transfer of the electron. You also have covalent bonds. Which involves a sharing of an electron pair. Sharing of an electron pair. If the sharing of the electron pair is equal between 2 atoms, call it equal sharing. The result is a more or less nonpolar molecule. That's what I have there. Non-polar covalent bonds. When a molecule as a whole, as a result, will be pretty inert and hydrophobic. Okay? Not very soluble in water. Non-polar, hydrophobic. Depending on the nucleus of the atom, the sharing is equal or unequal. If it's more or less equal, say for example in CO2, CO2 is pretty much a nonpolar molecule. Okay, and what it means in biology is it pretty much has no problem diffusing in or out of a cell. It can get past the hydrophobic zone easily because of its nonpolar. All right, so for example, if there's unequal sharing, say for example, the nucleus of the oxygen pulls the electron spinning around it closer to it. You have an unequal sharing. So still on covalent bonds, but continued if it's unequal sharing. It creates what's called a dipole moment. Kind of an odd term. I think what this essentially means is that the electrons spend more time spinning around one atom than the other. In this example, they spend more time spinning around oxygen than the hydrogen in a water molecule. So it creates a polar molecule. Say polarized molecule. Well, water is always a great example. H2O, where the electrons spend a little more time spinning around the oxygen, so the oxygen becomes not a full negative charge, but a partial negative charge, noted by delta negative. And so then the two hydrogens are partially positively charged, positive. And so in water, in a liquid medium, all the H2O molecules, they're attracted to each other by weak hydrogen bonding. So this partial positive charge is weakly hydrogen bonded to a partial negative charge of another nearby water molecule. Okay, and that negative charge there is weakly attracted to the partial positive charge of a hydrogen atom of a neighboring water molecule. And that creates a surface tension. Mosquitoes, or what is that? I don't think it's a mosquito. Water spider, yeah. It doesn't break the surface tension. It's so light. But there is tension. There is an attraction between the water molecules. Water is a great example of that. Okay, so hydrogen bonding is important. Water is a hydrogen bonding environment, as an example, as I've illustrated there. Let's move on. So this kind of like begs the subject of polarity. You have molecules that are polar, right? Unequal sharing. You have molecules that really are non-polar. They're inert. They don't have a lot of polarity at all. And so you kind of have to kind of understand the spectrum here. Ionic bonding, that transfer of electrons, that's kind of on one end, right? Fully charged ions, versus these kind of like polar covalent bond like water in the middle, right? And then nonpolar. Okay. So kind of to distill it down into two categories, are you polar or not? Because if you're polar, usually molecules have these kinds of atoms in it, nitrogen, oxygen, sulfide, fluoride, that really pull electrons close to it. And in biology, proteins, glucose, water, these are examples of polarized molecules that are hydrophilic. So polar is hydrophilic, nonpolar, hydrophobic. Things like fats, lipids, triglycerides, steroids with the complex ring structures, right? Things that are polar, things that are nonpolar. Even within amino acids, there are some kinds of amino acids that are more polar or less polar. For example, I would just say, answer my own question, which group is the most hydrophobic? I would say the orange category, because it has a lot of hydrocarbons, a lot of ring structures that make it hydrophobic. And that's kind of important to know generally speaking, okay? When you take biochemistry classes, you talk about it more. But just kind of get the concept of a molecule being polar or nonpolar. For example, here's a phospholipid. And what I had said was that phosphate group was polar. And that's important to know as a biology student because polar molecules can be soluble or interact with water. This end is nonpolar. It's hydrocarbon. It cannot interact with water. That's why the lipid bilayer for cell membranes, those tails face each other, away from water. They're hydrophobic. Okay, so all this chemistry for what? Why do they have a chemistry chapter in A&P? This is how you're going to apply it. So I want to give you a preview. Well, it's on your test, but it's also a preview for 431. How does all this chemistry apply to human physiology? So I'm going to give you 4 examples here. 4 quick examples: structure of water pores, and a couple examples in the kidney and in the lungs of how chemistry applies. Okay, so here's a picture of a water pore. I guess for a med school book. It's quaternary structure. It's got all of these. It's a trimer, okay? And each of these is 16 anti-parallel. I don't have to worry about that. It's the beta sheets, right? So the beta sheet was what level? 1, 2, 3, or 4. The beta sheet was 2. But what you can see is the third level is a bunch of beta sheets in a row form, say, this green one. Okay? But then when you link them all together like that, what level of organization is that? 1, 2, 3, or 4? 4. Yeah. Here's aquaporin. This is the water pore in humans. Okay? The level of organization is quaternary. It's a tetramer. 1, 2, 3, 4. That little spot right there is where the water can go through. That's the axis it can travel around. So let's note that. Aquaporin is important. Aquaporins, by the name, you know what it is. It's for water, aqua, right? Porin means that it's got a pore in it. It's a water pore. It has a quaternary structure. And they function in kidney cells. Let's give you a picture of that shown here. Well, I'm not going to describe the events in detail here. That's more for later on. But I want to give you the idea of how a water pore works in human physiology. That part we can learn now. Here's a cell in the kidney. Kidneys produce urine. I think people know this. What you may not know is it's producing urine by filtering your plasma. And we call that the filtrate. So on this side of the cell, where it's yellow, is the filtrate. The thing about filtrate is, whatever's in the filtrate that the body wants to conserve, you need to pull it out of there, back into the bloodstream, or you lose it to the urine. Because, once you urinate, it's gone. You void your bladder, you excrete all the waste, it's gone, you've lost it. So there's a lot of things in the blood you want to conserve, like water. So what they show you here is a blood vessel on the other side, a capillary blood vessel. One thing you want to conserve is water for fluid balance. You don't want to lose all the water being filtered to the urine, right? You want to conserve it. So if there's a water molecule here, it can't get through the cell membrane on either side. So it needs the aquaporin to be inserted on, I'll just kind of like insert it on one side, boom. So by rules of osmosis, water will flow, little blue balls, into the cell. It's got to flow through the other side. Osmosis is the diffusion of water. Well, basically, no. What I'm showing you here is you're reabsorbing water from the filtrate and you're putting it back into the blood. Reabsorb water to conserve fluids. You can only do it if the aquaporins are there. Okay, so you're taking fluid out. And so the kidney is something students can relate to. I mean, we urinate every day. If the aquaporins are not there, for whatever reason, your urine output is going to be a lot because you can't reabsorb the water. You're losing a lot of urine, okay? Maybe you're overhydrated. You're just kind of losing a lot of fluids that way. If you're dehydrated and you have a lot of aquaporins, this is happening, and your urine output decreases, and your urine is very concentrated. It's a very deep yellow color. But anyways, all that to say, the aquaporins are water pores. Other things that you learn in chemistry in terms of acid-base balance. In chemistry, you kind of learn how acids dissociate. If it's a strong acid like HCl, it completely dissociates in aqueous solution. It only partially dissociates for a weak acid, right? Some dissociate, some don't in a weak acid. In chemistry, this chemistry in the kidney, acids and bases are important for blood. Here's the same picture. Let's kind of draw it again. Here's the kidney cell. Here's the filtrate. Here's the blood. The pH of the blood plasma is very important in physiology. And you learn about pH in chemistry, right? What's neutral pH? 7. Okay, really that's 10 to the -7, right? Like molar. And the 7 is the pH, right? It's like the amount of hydrogen ions. So in human blood, the pH is around 7.35 to 7.45. That's the normal range that needs to be homeostatically regulated. And the kidney can help, okay? Let's say the pH of the blood, for whatever reason, gets too acidic. We call it acidosis. Let's say the pH becomes, for whatever reason, 7.25. It's below the normal range. That means, oh man, too much, the blood is getting acidic. So the kidney can help compensate, right? So what the picture shows is there's the amino acid glutamine. An amino acid, right, one of the building blocks. You just kind of break it into pieces. You just kind of metabolize it, and basically what they're showing you is, on one end here, you're going to secrete into the filtrate an ammonium ion, NH4+. Remember that? That's an ammonium ion. The ammonium ion, think of it as NH3 with an extra hydrogen added onto it. So what you're doing is you're acidifying the NH3 and you're tagging on another hydrogen to get rid of it, because you're going to excrete this in the urine. Get rid of it. And what the cell is doing on this side is you're secreting a bicarb, HCO3 with a full negative charge. That's your base, right? And you're putting that in the blood. So not only are you excreting acid, you're putting more base in the blood. So think about that. If you're adding base, what's going to happen to that number? It's going to go up or down? It's going to go up. If you add base to something, it's going to like increase back within the normal range. So, you know, I'm trying to get you to think about chemistry in the body. So that's another thing we talk about. The last thing I'll mention as an example is in the lungs. It's called external respiration. But look at this equation on the top there. There's CO2 there. Okay, so I'm going to draw this equation right there in reverse. It's kind of going that way. What this figure shows you is a couple of things. It shows you how you're able to breathe off CO2. CO2 is a waste product that builds up in the body. And it's the job of your circulatory system to get it to the lungs so you can breathe it off. So here's how you do it. You transport that CO2 in this form here. This is bicarbonate. Very important in biology. When it combines with a hydrogen ion, you form carbonic acid. You should still remember this from chemistry. And this can dissociate into CO2 and water. Okay? It happens really fast on the red blood cell because of an enzyme, which I talked about today, called carbonic anhydrase. I'll just call it CA. Carbonic anhydrase. Basically, speeds up the reaction. When you get the molecule in the form of CO2, you can breathe it off with each breath out. That's chemistry in the lung. So yeah, that's kind of my spiel on how chemistry occurs in the body, just to give you a preview for that. Okay. All right, so on the schedule today, we're going to start chapter 3. This is what it happens when you sign up for a 5-unit class. You move fast. If any of you are thinking of taking me for summer for 431, it moves even faster. But again, I'm only moving this fast because we had Monday off. This kind of makes up for it. Chapter 3 is cell, and I'll start off by teaching the cell membrane. In the cell membrane, you have fluid on the outside, the ECF, fluid on the inside, the ICF. The ECF is also called the interstitium. So if you draw a simple cell with the nucleus, the cell membrane, on the outside ECF, on the inside ICF, 2 fluid compartments, inside, outside the cell. Now the cell membrane has complex structure, so membrane lipids. The main thing, it's a phospholipid bilayer. That's the main structural feature. I defined earlier this morning, there is a hydrophobic zone, something that will stabilize the cell membrane because it's a very fluid structure. Hard to appreciate that in a still picture. There's these cholesterol molecules there. Cholesterol is a stiff molecule that helps stabilize, makes a cell membrane part more stable, less fluid. So I'll just put cholesterol. Cholesterol stabilizes. There's a lot of membrane proteins, right? The ones that I talk about there. Earlier this morning when I defined the channels, proteins, and pumps, things like that. Channels, transporters, pumps. They have membrane carbohydrates, all the green stuff illustrated on the surface, on the outer surface. So let me put glycocalyx. A glycocalyx is like a phospholipid molecule. Remember when I defined phospholipid? I said you had 3 carbons at position 1, hydrocarbon chain. Position 2, hydrocarbon chain. For a phospholipid, you have a phosphate group, but for a glycocarb, instead of a phosphate, you would have a carbohydrate instead. I should use, no, I don't have green, but well, where I drew it blue, they have it green. That's glycocalyx. These actually help in the function of cell identity. It's kind of like a fingerprint for the cell. The identity of a cell, well, basically, you don't want your white blood cells attacking yourself, okay? The glycocalyx can help white blood cells identify self from non-self cells. Self versus non-self. It's an immunology concept, but cell identity is the thing. So that's kind of like the long and short of it. And the thing about cells, they have to be attached to other cells to form a tissue. And I'll cover this in tissues as well. You have tight junctions, Desmosomes, and gap junctions. So let's kind of talk about each of those. So this figure is teaching you how cells can interlock with one another. So on the left, the tight junctions, in the tight junctions, the interlocking proteins, they like zipper the cells together. They're tight, so things can't pass between them. So I'll just say interlocking proteins zip cells together. I mean, just remember, tight junctions are tight. It's hard to slip through. And that's what they have illustrated at the top of the cell here, in this tissue. Right there, these tight interlocking proteins there. Also, you can have this interaction called the desmosome. The desmosome has an interesting structure. You have the cell membrane here in blue. But then you have these interlocking proteins I'll draw as orange. The interlocking proteins, they're like, well, they just call them linker proteins. An example is cadherins. They're like a Velcro that keeps the cells stuck together more or less. But on this surface here, you have a plaque that kind of provide a foundation for the cadherins to stick into. Drawing getting red. And then within those red plaques that I've drawn, you have other fibers that emanate out of it. Other thick and thin fibers that come out of it. And, well, These black fibers I have illustrated, they basically help form the cytoskeleton within a cell. So not only are these two cells linked together with the desmosome, the cytoskeleton helps provide structure for each cell itself. So let me see what they call it. Yeah, they say intermediate filament, keratin as an example. The function of desmosomes is like a glue that holds organs together. Like for example, in the heart, that's where I really focus on the desmosome. In the heart muscle, when it generates pressure, you don't want that pressure to tear the heart muscle apart. Desmosomes in cardiac muscle help kind of keep the cardiac muscle fibers glued together, so when you generate pressure, things don't get damaged. All right, so the last one there are the gap junctions. When the cells are bound together with gap junctions, the interlocking proteins called connexons have a pore through it, which allows communication because the connexon proteins are porous. That's the gap. The connexons have pores. So if there's a signal spreading through an organ and those cells have gap junctions, the signal will spread quite easily. Okay, make a note to yourself. Tuesday's lab has to do with this. I want to come back to these slides later. I just wanted to teach you basically cell membrane. It's hard to jump around.
On Tuesday?
I'm sorry. Okay, let me restate that.
Monday?
If you look at your schedule, Monday's lab is the microscope lab. That is what I gave you today. Okay. There's a lab on Wednesday called diffusion and osmosis. And so what I'm saying is the idea of diffusion will help get you ready for Wednesday's lab, the osmosis lab. And I want to come back to this like later. So let me be clear here so you know where we're coming and going. Monday lab. Microscope. Wednesday lab. Osmosis. Friday's lab. That's a DNA lab. Okay, I gave you that. I'll make hard copies for you for this. It's all on Canvas if you want to give yourself a preview, just so you know. Okay, so what I just said was, I want to pick it up here for some other time to get you ready for the osmosis lab on Wednesday. The microscope lab, to get you ready for that. For those of you that print out the slides, there's a batch of slides called, let's see, what is it? Mitosis. So any questions on the lab schedule for next week? It's on the schedule and I wrote it on the board. Okay. Okay, well, you know, I'm looking at this, I'm looking at the time. Let's just stop here. Okay, let's just do that. We'll hit the stop button here. I had meant to get through this